Hi, so this is the problem solving session 4 for basics of biology, and today we're going to continue our discussion on biomolecules. So, like last time, we had discussed the three of the biomolecules lipids, nucleic acids, and carbohydrates. In this class, we are going to start with our discussion on proteins. And then we're going to see what are the functions of proteins and what are the assays that you can use for detection of proteins and understanding their sequence. So we'll start with first what is a protein. So proteins are very large molecules composed of basic units called amino acids, which are joined by covalent bonds, which are called peptide bonds. And these proteins, they contain carbohydrates, hydrogen, oxygen, nitrogen, and sulfur. All these are constituents of the amino acids that form the building blocks of the proteins. And each protein can be broken into constituent amino acids by variety of methods. So you can break these amino acids separate from each other, and you can then analyze these amino acids in free form as well. And so to understand how does this complexity look like, so on your screen here on the you can on the bottom side, you can see that this is a protein structure. So this is how a protein would look with multiple chains, and this could be multiple motifs and domains and multiple units coming together and forming the protein structure, final 3D structure. Now you can see here that there are multiple helix structure here. These are the single structure here, loops like structures here. So this is generally called a tertiary or a quaternary structure. Now here, if you just zoom into one of these strands here, the helix here, and you try to understand how will this helix look like, it is something like this. So these are the multiple amino acids connected to each other via peptide bonds, and as you can see, these are differently colored. So this means that there are different amino acids. Each of these amino acids are different than each other. For example, here you have phenylalanine, leucine, and serine and cysteine. So we'll come to all of these amino acids in a moment, but you can understand that these amino acids are all connected to each other by peptide bonds, and this is how the chain will be made up. And to understand further how are these amino acids made up, this is the basic structure for an amino acid. So you see how the complexity keeps on increasing from a simple amino acid structure to a chain and then to this multimeric form. So let's start with the unit, which is the amino acid. There are 20 amino acids, which they all share a common structure, which is this CO and central carbon, which is called the C alpha carbon, and this is why they are all called. They're also all alpha amino acids because they have these alpha bonds here. And so, how are they characterized? So you will see this C alpha bonded to an NH2, to a COOH, one hydrogen, and one R group. Now this R group is what will give the characteristic to the amino acid. This R, if it is a polar, the amino acid will be polar. If the R is an aliphatic chain, then the amino acid will be an aliphatic amino acid. So this is how you you draw an amino acid. But if you also want to write the structure of amino acid, you can also write it like this. So you have to write you write C, you give this R here, take this H here. This COOH is how you write it if you don't want to show it in the bond terms, and NH2 is written like this. Now you can see that there are 4 different groups attached to this C. This makes this C or the C alpha as a chiral center, and this because we have read about chiral centers and stereoisomers in the previous session. So we can say that because of this chiral carbon, amino acids will have stereoisomeric forms. Now, as I have mentioned, that there are 20 amino acids, and all these amino acids are classified on the basis of their R group. So this side chain, they can vary in structure, they can vary in the chemical nature, and thereby they will have an influence on the overall property of the amino acids. And I've also mentioned that there are 20 amino acids, and generally when you write about an amino acid, we take the full name, but when you have to write it, you can denote it by a single letter or by the 3 letters taken together from its name. So we'll come to all of this in a moment. But just to understand the broad classification of the R groups are, or the broad classification of the amino acids are non-polar aliphatic, Aromatic, polar but uncharged. That means they are capable of forming a polar hydrogen bond, but in its native form they are uncharged. But then you have some negatively charged units which are negatively charged in their native form and positively charged in their native form. So we'll start with the aliphatic or the non-polar amino acids. So there are 7 amino acids in this group. It starts with the very simple amino acid, which is the glycine. So you need to focus on this group, all the colored groups here, which shows the R group, because the remaining three group functional group, which is the COOH, NH2, and hydrogen, that is common for every amino acid. So for the first amino acid, which is glycine, we write it as G or GLY for the glycine, and which only contains hydrogen as its fourth group. And this can tell you, this can give you an idea that it is a very small amino acid and a very flexible, because it's just an H group which is connected to the C. And on these all these snippets here, you will see that there are two values which are called pKa 2.34 and pKa 9.58. So to understand that, what you need to understand is that NH2 and COOH are capable of forming or getting protonated or deprotonated. Means they can acquire charge and lose charge as well. In its native form, they are COOH, but they can lose that H from the COOH group and become a COO minus. And they can acquire an H on the NH2 group and become NH3 plus. So that can have a negative and a positive charge. And why is that? Because they can these bonds, the OH bonds, are weak, or they are slightly electronegative, as we had seen in the previous lecture as well. So they can acquire negative charge here, and these values here are the values at the pH at which this kind of change will form. And when you have these kind of alteration of energy or charges, you call such kind of molecule as a Zwitter ion. Means it can have a positive and a negative charge. So when you see this kind of values, pKa of 9.58 means at pH 9.58 this can become NH3 plus, and at 2.34 this will become O minus. So that aside, so first amino acid for the aliphatic is glycine. The second is alanine. So this H now becomes a methyl, CH3. And how do you denote alanine? Again by ALA and A. Then you have valine. Now on the CH3, the two hydrogens are replaced by two methyls. So this becomes valine. Valine is represented by Val and V. I'll skip proline in a for a bit. The fifth one is leucine, which is again a modification of this side chain. And you see they are written by L and LEU. And isoleucine is an isomer of isoleucine. So there is just a rearrangement of these side chain. So what happens here is there are two H connected to the first C here after the alpha. So this one of the H gets replaced by a methyl, and that H is attached to the second C here. So there's an isomer of the or the rearrangement of the side group. That's why it's an isoleucine, and that is written by IL and I. And then you have methionine. Again, it contains all these C, but it is yellow in color because it is also important to note that it has a sulfur in its side chain. And it is written by methionine and M. And one of the important feature of methionine is generally the first amino acid to be translated or first amino acid to be coded for any protein. Now we come to proline. Now you see proline has this kind of a ring structure. What happened here is the CH3, CH3, one of the CH3 or CH2 formed a bond with the side chain or the amino terminal. So this becomes a ring-like structure, which gives this proline a very stable structure and a very rigid structure. And this is important because this will come up in a few slides, that because of proline, a protein gets like restricted to move around this axis. And it's written by PRO or P. Now the next group of amino acids are called the aromatic amino acids, and they are called aromatic because of the presence of these hexacyclic ring here, which can absorb at wavelengths, lighter wavelengths, and they can emit also their reflectance. So these have three categories or three amino acids. They have phenyl, alanine, tyrosine, and tryptophan based on what kind of rings they have. The thing to note here is that the single letter representative or single letter code for these amino acid do not really like follow the first letter of the amino acid. So for example, phenylalanine is represented by F here because remember P is for proline. So phenylalanine has F and it's denoted by PHE. Tyrosine is given by TYR because T, as we will see in the future slide, it is for threonine. And again, T here is again represented by, like, 3 is for threonine, so tryptophan is represented by W or TRP as its three-letter code. Then we have polar uncharged. As I mentioned, polar uncharged means they have the capacity to bind to form polar bonds, but in its native form they are uncharged. And these consist of serine. So here you can see that serine has SER as its triplet letter and S as its single letter code. And if you see the functional group, they will have a CH2OH. So this OH is what enables them to form a polar bond. Similarly, threonine also has an OH. Cysteine has an SH. Asparagine has this double bond CO and NH2. And glutamine has a CO NH2 as well. But the change is in the number of CH2 attached to this R group. And serine is represented by S. Threonine, as I mentioned, is represented by T. Cysteine is again colored in yellow. It means that this has a what do you say, like S, sulfur containing group. And glutamine and asparagine has an NH2 group. That's why they are orange in color. And they're represented by N and Q. And glutamine is written as GLN and asparagine is written as ASN. Now, next is negatively charged amino acid. That means they will have negative charge in their R group. Now, to note here is the name. So, you just came across these names in the previous slide, asparagine and glutamine. So, you can understand where did this name come from. So, this is CH2COOH, or COO minus. So, this is aspartate. If you go back to this slide and you see asparagine, you have here CH2, but CO and the other part is NH2. So that's why when you have NH2, you are calling it an asparagine. When it is only with COO minus means it's an acid. So that's why you call this an aspartic acid. Similarly, when glutamate has COO minus, when it will have an NH2, it will be called as glutamine. And in the other form, it is called either a glutamate or a glutamic acid, and they are denoted by different letters. So they are denoted by D and E, and aspartic acid is for ASP and glutamic acid for GLU. And the positively charged amino acids are these, the lysine, arginine, and histidine. They are very important. They are found in several of the pathways. For example, lysine, arginine, they are found in as several precursor for several of the biomolecules that are synthesized. Lysine also, you will also find them in ketogenesis. Histidine, you will find them to be structural component of many important proteins. So they are very important amino acids. And they are represented by K, R and H for lysine, arginine and histidine. So these are the 20 amino acids that you find. Now, there are some questions here which are from the previous assignments. So I'll read the questions and I'll give you some 10 seconds time to see if you can answer this. So the first is the amino acids which lack beta carbon. So beta carbon is all the other carbon in the R chain except for the one that is the central carbon. So the central carbon to the amino acid is called C alpha or the alpha carbon, and any other C in the R group is called the beta carbon. So which of these amino acid lacks a beta carbon? The amino acid which is linked with the thioester linkage. Thioester linkage is it's given, the name is given to something which contains a sulfur. And then we have the amino acids used in ketogenesis generated during the acetyl coenzyme A metabolism. And the last is the type of amino acids present within the interior core of the globular protein as well as on the surface of the transmembrane region of the integral membrane protein. So you need to understand that if you are on the surface of a cell membrane, you have to interact with the outside molecules, and for that you need some kind of bond formation. But if you are on the interior, you are shielded from all these molecules, so you are you don't interact with them. So the ones that would be in the interiors are the one that will be characterized by these kind of side chain that are not capable of forming a bond. So with that information in mind, try to answer this question. Third is which one was used in ketogenesis. So for that, we have leucine and lysine. And again, the ones which are present in the transmembrane domain are the ones which non-polar and hydrophobic. So you see, hydrophobic means they do not interact with any water. So they have to be in the interior membrane, and they're non-polar as well. So that's why these are the amino acids that will be present in the transmembrane domain or on the interior core of any protein. The next set of questions are amino acids with aromatic side chains are which of these following amino acids? Amino acids with the aliphatic R group. Again, you have to find out which of these contain R group which are aliphatic. The amino acid side chain most vulnerable to oxidative damage are which of these following? And the amino acid iodinated in thyroglobulin is which of these following? So the first one was easy. You just need to remember the three amino acids that were in that group. So we had phenylalanine, tryptophan, and tyrosine. The second question had the ones which are from the aliphatic. So there were seven amino acids, and you need to find out which. So let's go over each of them and try to like remember which one was which. So we go to the second first, which had serine, threonine, and cysteine. So because it was threonine was a polar uncharged, and cysteine was also a polar uncharged. So that's why we have this here. Yeah. Then you have lysine, histidine, and arginine, which were positively charged amino acid, and phenylalanine, tyrosine, tryptophan. As you know, they are negatively charged amino acids. So that's why the answer is glycine, alanine, and leucine. And then we have the amino acid side chain, which is most vulnerable to oxidative damage because they contain the positive charge on them. And oxidative means which is capable of binding to positive, which can attack the positively charged ion. And the amino acid which is iodinated in thyroglobulin has tyrosine residues which are iodinated. So they have iodine attached to, or iodine as a group attached on the tyrosine. Now, the next thing is we're going to read about how do we analyze amino acid by TLC or thin layer chromatography. Very easy technique to understand. So, this is called chromatographic technique, which is used to separate and analyze the free amino acids from any protein. And what you do first of all here is the idea is that there will be a solvent, there will be a stationary phase, and you will have your own sample. So if you just look here, this will be your chamber and this will be the plate. To just understand in basic term before we go into the technicalities and the details, there will be a solvent here or a mobile phase which will move across this plate or the stationary phase or the medium that you have given, and based on what is the affinity of the amino acid towards its solvent and the where does it stop on the stationary phase, you can identify the amino acid. So let's understand a bit more. So in this method, you have this silica or alumina which is coated on the stationary phase. So stationary phase means something which will not move. So this will be a plate on which you will be having a silica or alumina coating. And the plate could be of glass or aluminium foil or anything. But you have to make it as very thin, because if it is very thick, the solvent will get like will not be able to move very swiftly. So you make it as a very thin layer, and then you allow your sample to run on this in the presence of mobile phase, because by itself on the stationary phase your sample will not move. So you will ask the solvent to mix with the sample and start moving upward towards the on the plate, and as and when your sample stops, the position where it stops is where you analyze your column finally, or the paper or the pattern finally. And so what you first do here is that so you have this solvent, you have this sample spot. So the sample will run along with the mobile phase and it will get distributed in the solvent phase and stationary phase. And the interaction of the samples with the stationary phase will retard the movement of the molecule. And so there is this two forces going on. So the components are experiencing two kind of forces. You have the mobile phase, which is trying to move the sample onto the stationary phase, and you have the stationary phase which is trying to stop the movement of the molecules. So you have something like this, where the mobile phase is giving a force which is trying to move the sample up the plate, while you have a stationary phase which is giving the retardation force which is trying to stop the molecule from moving. So wherever now, because the molecule starts by moving, your Fm, which is the mobile phase force, is always greater. Wherever your mobile phase uh, force will be equal to the force generated by the force exerted by the stationary phase, that is the position where the molecule will get immobilized, because there it cannot move any further as the opposing force is equal in the magnitude. And this forces, or where will it be immobilized, it is determined by multiple factors. The first thing is the molecule itself, so R group will determine how long it can run. And then the phases that you use, what kind of phases, is it completely miscible with your solvent, or have you made a stationary phase too thick so that the solvent cannot move across it, or what is the, in the stationary phase, which kind of stationary phase, is it a silica, is it aluminum, is it reacting with your solvent, is it reacting with your sample? All these factors should be taken into account when you are doing this kind of analysis. So once you have your run completed, this is how it will look. So how do you calculate what is the retention factor? So if you try to understand, this is the plate, this is where the solvents will move, and each of these columns here will be the spots for each sample. So the first thing that you calculate is how long has the solvent moved, and that you will be calculating by looking at the solvent front. So you need to be very particular of how long you are running, and after that point of time, you need to see where does the solvent front stops. So you calculate that distance, and that distance is the denominator for every retention factor that you calculate. And then what you do is you calculate the distance moved by the sample from the uh, center. So the center of the spot is divided by the distance traveled by solvent. So for example, A here. So let's consider this to be 5, and let's start with A. So A has moved this much distance and this is 2. So the retention factor for this spot here will be 2 by 5, which is 0.4. Similarly for B, which has moved 3, will be 0.6. For C, it will be zero point 0.8 by 5, and for D, it will be 4 by 5. Now U has two components. This is also possible that your sample has two kind of components, and those each individual component has individual retention factors. So you calculate retention factor for each of the components separately. So for U1, if you consider this to be U1, this will be 3 centimeter movement, so 3 centimeter by 5, and for U2 it will be 0.8 by 5. So that's how you look for your retention factor. And once you have a retention factor, you know your solvent, you know your stationary phase, and you have the retention factor. Taking all these factors into account, you can identify which component it is. So let's just take you over the step. How do you actually conduct this experiment? The first thing is spotting. So after you have made your plate, you make this first line here, which is called the spotting. Now this spotting here is important because this will be a start point for you to refer to where you have given your samples. If you give a sample, you lose your baseline. So you keep your baseline a little above the plate bottom, and then from here you load your samples. So that's the second step, where the sample is loaded into capillary tube and you make a spot here, and you allow the spot to dry because you want it to move with this solvent, with the mobile phase. So you allow it to dry, and then you run it. And how do you do that? So your spot is dried, you place it in the TLC set chamber in such a way that the spot should not be below the solvent level. The solvent front is allowed to move until the end of plate. So you keep noticing your solvent front, it should move till the end of this plate, or at least somewhere here. Once you are done with the experiment, you can see in this picture here that this sample that you have loaded from this spot has started to open up, and it had multiple components. So each component is resolving, moving at its own pace in the solvent. Now this is where you have the end result of your TLC. So this is from, I have taken from internet, and this is one of the picture from our lab. You can see how these different components resolve in the TLC plate. So this plate then you take out of the chamber, again dry it, and now there are three ways of analyzing it. If the sample itself is able to give you color, then you have these color spots. You can identify these different spots. You can visually look at it and you can try to calculate the retention factor. But if you don't have any such spot which are visible, then you have to do additional steps. For example, you stain them. So you can stain them using Forex or ninhydrin or anything where you can make out the spots which are formed on the TLC plate. Or if you don't want to stain them, you can do autoradiography or fluorescence if your compound is able to fluoresce, and you can find out where exactly the spots have formed. And then you take forward the information of all the compounds together and try to identify which amino acid or which protein it is. So based on the topics that we have covered so far for TSC, these are some questions. So let's try to answer that. So the first is, in thin layer chromatography technique, the phase retards the movement of the molecule during the interaction of sample is. So which phase retards the movement? Then you have, which of the following is not correct regarding thin layer chromatography? So does it is it based on, so you remember it is not correct, so which is incorrect. So it is based on adsorption mechanism. It depends on the functional group present on the analyte. The retention factor is unaffected by temperature and layer thickness. Polar compounds will have a low retention factor than non-polar compounds under some developing conditions, same developing conditions. And the third is, which is not an application of TLC to isolate vitamins, to identify drug, to act, to isolate neomycin sulfate, or to determine moisture. And the last is, which is not a requirement of the mobile phase. Should it be not toxic? Should it be easily available? Should it be chemically inert, or should it be chemically inactive or active? So the first question was about the phase that gives you a retarding force. So which was easy because it was stationary phase that retards the movement of the sample. The second was, which is not correct about TLC. So the retention factor is affected by temperature and layer thickness, and we have spoken about it. Then which is not an application. So you can identify, you can isolate, you can identify drug, isolate vitamin. All these are components which can resolve in the TLC plate, but you do not test for moisture in TLC because the idea is to separate your compounds here. And the last is, which is not an important requirement for mobile phase. So it should not be it should be chemically active is what is wrong, because you don't want it to be a chemically active necessarily. Okay, then we move on to peptide bond. So remember, we had spoken about how proteins are chains formed of amino acids that are connected to each other by peptide bonds. So peptide bonds are basically condensation reactions. So if you have to see here, so they are condensation reactions. And how are they formed? So let's consider these two amino acids. So this amino acid will have its C terminal COH and N terminal NH2, and the second amino acid will also have its C terminal COH and N terminal NH2. So the COOH and NH2 phases of these two amino acids will interact with each other, and there will be a loss of water molecule, and this C and this N will get bonded to each other, and then you have this bond which is called the peptide bond. So the peptide bond is characterized by a CONH bond, and this CN here is a partially double character because you can have these C and N interchangeably changing their charge, but this is quite a they undergo resonance, but and it is quite a stable bond. So if you see an amino acid, this is a very strong bond, so the amino acid cannot rotate, or the protein cannot rotate across this axis, but you have these axes that can move. So this was a C alpha. So let's consider one of these amino acids here. So this C alpha is the C from the chiral center of one amino acid. So this is one amino acid here, and this is C alpha. It has its H, it has its R, and then it has a CONH bond this side, and then it has an NH bond bounded to COs of some other amino acid. So there's a CONH bond this side and a CONH bond this side. So there are multiple kind of bonds here. First was the CN bond, which is called the omega bond. Then you have the C alpha, which is bound to the N, which is called the phi bond. And you have the C alpha bound to the C bond, which is called psi. So easy to remember. If you forget, it is psi means C bonds, and the other one is phi bond. Now these bonds, these are free to rotate, and these bonds can rotate. Means what? If you try to imagine as an axis like this, so these amino acids, they are free to rotate like this across its axis here. So these bonds can move in these directions in one plus 180 to minus 180. Plus 180 means movement in one side, and equally it can move the opposite side as well. Now these are important thing because this gives flexibility to your amino acid and then as a protein as a whole. So your proteins can move across axis, keeping your C and bond of the peptide bond constant. The other bonds can move around different axes to give different conformation to your proteins and different conformation to the amino acid as individual components. And this is what was seen, or it is what you do when you plot a Ramachandran plot. So what this Ramachandran plot does, so this is given by G.N. Ramachandran, who determined the possible phi and psi angles for a particular amino acid in a synthesizing tripeptide. Tripeptide means one amino acid here, one here, and one here, so that you have all these bonds together and your amino acid in the middle, so that you see if in bound structure how much this amino acid can move. And then based on these calculations, he constructed this plot which defines the region of allowed rotation for amino acids present in a protein structure and proposed to use this for validating a 3D structure. So what in this kind of plot, this is the minus 180 to plus 180 axis, and there are different kinds of secondary structure to a protein. So it can be a beta sheet, it can be a helix. We had seen helix, we can also have something called beta sheet. So these are for being in right-handed helix, a particular amino acid, the one which is, for example, for the one for which this plot has been generated, for being in alpha helix, these are the possible pi and psi combination that then amino acid can take. For being in anti-parallel beta sheet, these are the possible amino acid, or these are the space that it can occupy. And for the left-handed alpha helix, these are the spaces it can occupy. So that's how you interpret an amino acid. Now, these are the plots for different amino acids. So in general, when you see these are for different amino acids, but three of these are very characteristics. First is glycine. Remember glycine had H. So it is very, very flexible to move around. And I had mentioned this point when I was speaking about glycine. And you can see by the proportion of the phi and psi angles which are present all across the space, which gives you an idea that the angles of this phi and psi can change very variedly. Compare this to proline, which has a very restricted range for the possible phi and psi rotation, and this is because proline is a cyclic nature, because of which the structure is very stable. So they are conformationally more restricted than the general case. But then there are something called pre-pro. Pre-pro means residues which are preceding proline, but you don't consider prolines or glycines because they are separate, because they have their own plots. But if there are amino acids which are before proline, remember proline has this ring, so it gives the structural stability around itself. So amino acids which are present just before the proline will have more restrictions compared to the general cases, because in general cases you don't have any other amino acid or any other side group hindering your movement. So that's why in pre-pro residues you have different the possible combinations as compared to general cases. Now all this was about the amino acids, and amino acids make up the basic unit of protein, which is called the primary structure. So based on so similarly, protein has four categories of structure. We have primary, secondary, tertiary, and quaternary. So primary is just the linear change of amino acid, which will all be amino acids be connected to one another. Secondary is the first layer of twisting and folding this amino acid chain undergoes, and that gives rise to alpha helices and beta sheets. Third is when these helices they wound around each other to give a 3D structure, which will be composed of these beta sheets and alpha helices together. And the fourth will be the quaternary level, which is different tertiary structure coming together. So they can have different chains of amino acid coming together, or different uh, peptide units coming together, protein units coming together and forming the quaternary structure. They can have multiple subunits in these cases. So primary structure is where it's a linear structure of amino acids. So the order of the amino acid determines the folding of the protein to achieve net minimum free energy. So any conformation in any uh, native form in the nature will always try to reduce its free energy. So of the order of the amino acid determine which is the uh, net free energy, net minimum free energy of the conformation. And how do you do that? You do that by multiple permutation combination, looking at which of these folding and will be the least energy one, and then you will retain that. So now this was about protein structure. You can also determine what amino acids are connected to one another by several methods. So you can determine the primary structure. We had seen to analyze the amino acid, but to understand which amino acids are connected to one another, or which amino acids are connected in the series of the peptide chain, we have several methods. But to do that, the first step will always be to break open the protein. And by breaking off of the protein, so if you have disulfide bridges. Now, what are disulfide bridges? If you have two S's in the amino acids and the chains, these SH can form a bond with each other by losing the hydrogen and forming a disulfide bond. These disulfide bridges are where the proteins are extremely stable. For example, you see here, this is one chain of amino acids, this is one chain of amino acids, and you have these two S's facing each other. They will form a bridge with each other, making this protein very stable in structure. So the first thing is you need to first break off such kind of disulfide bond to get to the individual chain level. So the very first step, if you want to do any protein sequence determination, is to break the disulfide bond open, as that interferes with the complete sequencing procedure. And to break that off, you have two ways. The first way is to oxidize it by so you oxidize it with an acid to produce cysteine residues. So you break open the sulfur bond here, and then because you have an acid, you add these OH groups to them, and they become cysteic, so cysteine acid groups. So the either one, by using any acid to oxidize these bonds here, or to oxidize sulfur here, or you can use something called DT3, dithiotrietol or mercaptoethanol, where again these two cysteines will form a carboxymethyl cysteine. So here, if you see, if you use this DDT, so the S will form a COO. They will acquire a carboxyl group. So they will become a carboxyl methyl cysteine, and they will inhibit the formation of disulfide bonds. So you add a group onto the sulfur to stop it from binding with each other. So that's the first step. You first break the disulfide bonds present in the protein structure. Then you break the polypeptide chain. So you start breaking each of these component here. And how do you do that? So there are several like amino there are several proteins or chemical agents that can break off at different amino acids. Remember something like this we did in Sanger sequencing for DNA sequencing, where we had piperidine and hydrazine. These compounds were able to cleave either guanine or guanine and adenine or thymine at particular sites. So similarly here also we have some proteases or some chemical reagents that can target the protein by recognizing the sequence, and they can cleave after a particular amino acids. And what are these? For example, you have something called aminopeptidase. So aminopeptidase breaks between CO and H bond, which gives in the name itself, aminopeptide days. Then you have pepsin, will also cleave at the CONH bond. And then you have chymotrypsin, which will cleave after this kind of aromatic group. Pepsin will cleave after this kind of aliphatic group. Trypsin will cleave after and will positively charged amino acids. So, for example, lysine or arginine, it will cleave after that kind of amino acid. And then you have carboxypeptidase, which will be cleaving off at the CONH bond with the two with the which between the two amino acids. Either these, or you have something called thrombin, which can cleave if there is an arginine lysine bond, so it can cleave between the arginine and lysine. Or if you have cyanogen bromide, then it will be cleaving at the methionine. So it will cleave methionine and give rise to something called homoserine lactose. So you have these different reagents that can cleave at different amino acids. So now you have generated peptides. First you have cleaved the disulfide bonds. Now you have individual chains. You have cleaved the chains as well. So now you have smaller peptides. So now you will sequence each of these peptides to understand what is the sequence of amino acid in them. And for this you have again two methods, which are Sanger and Edmond degradation. Now for the Sanger method, Sanger method is based on what do you call as the identifying the N-terminal residue. So here also we start, remember, for again with drawing corollary with the DNA sequencing, there we were labeling the 5 prime end so that we understand the starting of the sequence. Here we will start with identifying the N-terminal residue so that we move from N-terminal to C-terminal. And how do you do that? First thing is you form a derivatized form of the N-terminal residue. So what you do is you label the terminal amino group on the end side and N terminal with compounds like DFNB and densyl chloride. So you have a tribe, suppose you have a peptide, and then you have this compound here, and then you ask it to label it. So in this end, in the NH2, this group will be added. Now, next is you do acid hydrolysis of this compound, and now that because of that, you will have the breaking of peptide bond, which will release this dinitrophenyl amino acid complex. So if you now hydrolyze this protein, the bond will be broken as such that you have this group with the first amino acid out, and then the amino acids are remain as free amino acids, and then you can have these free amino acids to separate on the TLC. So what you do is separation and analyze the derivatized amino acids in the HPLC or TLC as we have gone through, and see which are these amino acids. So you have this one, you can load onto this and you can see which amino acid will stop at that spot, given that you have a DNP attached to it based on its weight. Okay. The other form is again based on N-terminal sequencing, which is called the Edmond degradation sequencing. Here also, you start by reacting phenyl isothiocyanate with the terminal amino group to form a cyclic phenyl thiocarbamoyl derivative. So you have this amino acid here, you have the PITC group attached, and you form a derivatized protein. Then again you give an amino acid so that you cleave the amino acids terminal group here. And then, so when that happens, it forms something called thiazolinone derivative here. This derivative is extracted into organic solvent, and which forms a phenyl thiohydantoid amino acid derivative, which you are basically rearranging the compound into another compound which is amenable to HPLC separation, so that by HPLC separation you can see which one is this compound so what happens is, in HPLC separation you have an amino acid profile. There you compare this with the standard amino acid and say that, okay, if I add PTH to this amino acid, how will it look? And based on that, whatever profile you find, you try to identify the amino acid. Now this was N-terminal, one amino acid. You have the free peptide with another N-terminal, which was originally the second amino acid. Now again, this chain will give you this kind of amino acid at the second round, with which you can identify the second amino acid, and this chain will continue till you reach the end of the protein or end of the amino chain sequence. Now that's what you do to identify the sequence of the protein. So these were from N terminal, but C terminal residues sequencing is, for that we don't have many of the methods which are well developed. So once you have got the amino acid sequences, then you order them. So you have different peptides. Remember, you had, this was from one peptide fragment. So you have different peptide fragments, and you have these different peptide fragments overlapping with each other, and you will have amino acids which will overlap with each other. With that, you try to form the sequence. So for example, let's give you an idea here. So let's say this was one of the peptide fragment, this was another peptide fragment, and this was another peptide fragment. So what do? You get some sequences like A, and then say G and M and P probably. And then for the another peptide fragment, you have some M, P, and then some C, S, and T. But for another amino acid fragment or another peptide fragment, you have something that starts with ST and then A, R, and P. So you have three amino acids, three fragments which are giving you three amino acids combinations. So this is the first fragment, this is the second fragment, and this is the third fragment. So if you now want to look at the original sequence, you will start moving across this. So first you will have AG from the first fragment. Then you have MP, which is present in both the fragments. Then you move to C, S, T, and then you add to this A, R, and P. So this is the amino acid sequence for the entire protein, which you have generated by sequencing the individual peptide fragments. And once you are done this, then you try to go to the folded form by locating the regions where possible doubles the disulfide bond could have been present. So that's the idea, that you go from the linear structure to the complex structures. The other method, or the method that are now prevalently used, is called the mass spectrometry method. So what you do here is you have mass spectrometry in conjugation with proteomics information, which you use to deduce the amino acid sequence. So very briefly, what you do here is you have the peptides. So you have the peptides here with some charge to mass to charge ratio. And first you analyze them. So you have these different mass to charge ratios. Now you pick any of the fragment from these so here also, you will first cleave your protein, you will break the disulfide bonds, generate peptide fragments, and then run these peptide fragments in mass spectrometer. So you get the such kind of spectra, which gives you the mass by charge ratio of each of the fragment. How much is the mass of the fragment is constituted by how many amino acids it will have. So you have these kind of mass spectra. Then you select one of the fragment and you further fragmentize it with in a CID, and then now it will be fragmentized. So here that peptide was very small, in the sense it was a part of the total protein. Here again there will be some bonds will be broken, and then you determine the m by z ratio of the peptide fragments in the second mass analyzer. So there you have these, again you have fragments generated, you will have these kind of fragments. So you then determine that, let's say you have these peptide fragments, Then you do bioinformatic analysis to identify the protein based on the amino acid sequence of the peptide with M by Z 1395. So you have this kind of pattern. You try to see which kind of protein will have this kind of possible spectra given that you have fractionized it. So you use some bioinformatics and you use some proteomics tool to identify the possible kind of amino acid sequence, and then you try to see which kind of protein is possible. So that was all about what you do for protein identification. But we move on to some folded structures now, which are secondary protein structure. So secondary structures will generally mean that the amino acid they interact with each other, and as a result, the peptide chain will now fold into secondary structure, and this will give rise to further tertiary structure. They are of three types. Basically, they are of alpha form and beta form. So you have alpha helix and beta sheets. So the alpha helix first. So in this, you have the polypeptide backbone wounded around the central axis. So there would be, so let's consider this to be a amino acid chain, and this consider this to be a what do you call central axis, and then you have this chain moving across this helix. So you, moving across the center axis. So it will keep rotating and moving across the axis, giving rise to helix form. And the R group of the amino acids will all keep protruding outward from the helix backbone. And if the bounding is in right-handed direction, the helix is right-handed helix, and if it is in the left-handed direction, it is called the left-handed helix. So you can see here that if you consider this to be the central axis and this to be the straight chain amino acid sequence, they have started wounding around the central axis with the R group projecting outward from the interior. So this is the alpha helix structure. And how does that happen? You have these interactions between the bases or the amino acids groups, which will hold them across the central axis. So that's how you give rise to a helical structure. The other is a beta sheet. So this is more like an extended form where the R groups are again protruding from the zigzag structure in opposite direction, giving an alternating structure. So you have these sheets. So you have these structures to each other, and let's say both of them are facing the same facing. So both of them are C and facing, and you have these R group and one of the H group interacting with each other. So you can imagine that there are two lines going one beneath the other, and they are connected to each other. So they will be forming like this kind of structure. If you fold a page multiple times and then you open from one end, they will have these different pleats like structure. That's exactly what you call a beta sheet. Now just try to imagine that the individual lines that you see in the paper, if they are parallel in nature, then this is a beta sheet, which is a parallel sheet. If they are anti-parallel in nature, then they will be anti-parallel sheet. So here you can see that if they are parallel to each other, these the strands are connected by these kind of interaction or these kind of structure. If they are anti-parallel to each other, then they will be connected by the NC terminal. So if you consider this C connected to N and this C will be connected to the next N. So that's how you get connected to each other. Now you have turns. These are secondary structure. They will not have without any definite structure, but they are present in protein structure to change the direction of running. So you can imagine that these are called turns because they are rotating the chains upon one another. So they can either connect two beta sheets with each other or one helix with a beta sheet. So you can see if you want to see beta turns here, you have these ends, okay? And the CONH bonds are here. Is R1, R2, R3, R4. So ideally this would have been a single straight line, but what happened was for the the C, the first amino acid, the CONH, the CO bonded like it attracted the H group from the NH2 terminal, N terminal of the fourth amino acid. As a result, the fourth amino acid and the first amino acids are now connected to each other, protruding the third and fourth out from each other. So this part here forms a loop or a turn, and the R group of these are facing outside. So that's why this is a turn here. Similarly here, so you have a beta turn here as well. Now, why is this alpha and beta? Again, based on what kind of orientation the carbonyl group will have here. CO at the bottom or facing the interior side is the type 1 kind of turn. CO facing outside is the type 2 kind of beta turn. Now, moving to tertiary protein structure. So, a secondary structure folding on itself will give rise to a tertiary structure. Something like this. So, you have these chains. Now, they will start interacting with each other via hydrogen bonds or disulfide bridges or ionic bonds. And then you have quaternary structures which have multiple polypeptides coming together and for giving rise to the functional unit of a protein. For example, hemoglobin, they are heterodimers. So they are basically alpha and beta, two dimers. They are themselves, first of all, of two units, alpha 1, alpha 2, beta 1, beta 2, and all these four come together to give rise to the hemoglobin that is present in your blood cells. So that is what we call quaternary protein structure. And these structures, they are determined by the use of some experimental methods, for example X-ray crystallography or NMR spectroscopy, or you can also do them by something called homology modeling. So you have these sequences, so you can use the sequences, go to a database and check that if there is this kind of sequence possible, then what kind are the possible conformation that these amino acids or these proteins can take. And from that you can try to imagine that, okay, there are certain set of proteins which have already been found to have this kind of sequence and this kind of structure. So this amino acids sequence from my protein will also possibly give rise to this kind of structure, but given the fact that it will have a minimum free energy. So that's how you can try to imagine the structures. Now, based on the discussion that we had so far, I've compiled these questions from previous year's assignments. So the first is, the reagent used in the cleavage of polypeptide change after our aromatic amino acid is which of these? Uh, the method used to determine 3D structure for the target protein, again we have just discussed this. The structure solely stabilized by covalent bonds. So we just spoke that if you're changing your structure, what kind of bonds come into play, but you have covalent bond only. So which kind of structure will be important for it? And the major driving forces for protein folding are, so these are different interactions that take place, but which is the major one. So the first one is for chymotrypsin, as we had seen in the previous slides. The second was NMR. The third is primary structure, which has covalent bond, which is the peptide bond that is stabilizing them. And the last is the hydrophobic interaction, because you remember they are called protein folding. So there will be the sequences which are, these are the interactions that are responsible for getting these chains to close to each other. Now we move to protein functions. So all these are, we have read about proteins. What are the structures? What are the possible kind of conformation that it can take? We will now look into the functions of protein, or what kind of function it can undertake. And that is actually determined by the 3D structure that it takes, because in that form the R groups are outside, which of the groups are facing inside will determine which of the possible amino acids that are there available to interact with other residues or other biomolecules. So based on that kind of structure only, the function of the protein is determined. And there are several functions that a protein can undergo, and these are just some of them which are listed, and we'll go over each of them one by one. So the first is enzyme. So enzymes are basically biological polymers that catalyze a reaction. These proteins have catalytic capabilities crucial to perform different processes, and they can enhance the rate of reaction as high as 10 to the power or 10 to 17 folds compared to uncatalyzed reaction. They react with the substrates here, for example. So these are enzymes, they have substrate, so they react with the substrate and convert it into products. And they are we have already seen so many examples where these are very important, because they help to move forward a particular reaction, and they are part of step by step reactions. The previous step gives rise to the next, or the previous step product is what is used as a substrate for the next step. So they are very important for sustaining life. And you also have some RNA which act as enzymes, which are called ribozymes, and they act on themselves. They catalyze their own reactions, and that they are also, but the idea is still the same. So in a very briefly, to summarize all these points, enzymes are biomolecules or proteins that will be capable, or that will enable a reaction to go at a faster pace. Just try to imagine a sugar converting to glucose, a sugar that you have in your house anywhere. If you keep it open, the sugar will convert to glucose or hexose in some day or a time. But if you provide some enzyme there, enzymes is, their function is to attach to the glucose and or sugar and convert it to its monomeric form glucose. And what will it do? It will reduce the activation energy. So the compounds need a certain energy to activate itself, to transform into some other component. By itself, if you leave the component, it will take many years probably to come to that activation energy, by which it is very low for it to undergo a reaction. Enzyme will bind to the substrate, or in this case the sugar, and reduce the activation energy at such a pace that the what was happening in years will now happen in minutes. So the activation energy goes down, and as a result the reaction moves faster, at a faster rate. So that's the idea of enzyme, but that's why you use an enzyme, so that you catalyze this reaction at a faster pace. So that's what you're showing. The enzyme's purpose is solely to bind to the substrate, forming a complex, give out the product, and release itself and be ready for another round of application. There, and based on what kind of substrate it binds to, what kind of product it leads to the formation of which kind of products, they are classified in these different type of forms. So first is oxidoreductase. Name says, so it is it catalyzes oxidation reaction, where the electrons tend to travel from one form to the other, and they are reducing, because the electrons are reduced going from one form to other, they are reducing the substrate. So there's an oxidation reaction which is carried forward with the reduction of the substrate. Then you have something called transferase. These enzymes, they transport the functional group of among acceptor and donor. So they take one group from the let's say one acceptor and give it to another donor, or one group from the donor and take it to the acceptor. So they transfer the groups from among the protein, or among the molecules. Then you have hydrolases. So if you have something called ases, that gives an idea that this is an enzyme and it is catalyzing a reaction. So hydrolases is for hydrolytic enzyme, and these catalyze the hydrolysis reaction. Hydrolysis, using water, you're cleaving something. So you are taking water and you're cleaving the bond. So basically you are hydrolyzing it. Why? Because you're adding water to it. Then you have something called, some classes called lyases, where you add water, or the enzymes add water, carbon dioxide, or ammonia across double bonds, or eliminate these to create double bonds. So it either generates a double bond or it breaks double bond by adding these kind of groups. Then you have isomerases. Isomerases, they cause this shift, structural shift. So the rearrangement of the molecule, and because of which there will be a change in the shape, and shape is a very important determinant in the functionality of a biomolecule, as we have read so far. So that's why these enzymes are important. And then you have ligases. So these will catalyze ligation. Ligation means joining. So if you have, let's say you want to join two products. So let's say you want to join DNA and a vector in the process of cloning. Then you have ligases which will attach these two components together and to form the insert of your cloning vector in the process. So that was the first function, which is enzymatic functions. The next one is where it binds reversibly to a protein, or reversibly binds to a ligand. What it means by reversible binding is that it will bind to the ligand, but it will that's not a permanent bond. It will release the ligand as and when required. And a very important example of this is the hemoglobin, which carries oxygen. Now where does it carry oxygen, where does it release oxygen? So it carries oxygen, it is in the red blood cell, and it has to deliver oxygen from the lungs to the tissues. So it will bind to oxygen at the lungs, move in the body, and at the tissue sites, it will release the oxygen. So that's the kind of reversible binding that we are talking about. And it is not just with oxygen, you also have carbon dioxide transport by reversible binding. So gaseous oxygen is, as we know, that it is required for transporting from atmosphere to the inside of body. And we have hemoprotein, which are hemoglobin or myoglobin, which have the active site where the oxygen can bind. And these hemoglobin, they are present in the RBC, and at the lung surface they can bind to the oxygen carried in the body, release in controlled manner to provide oxygen for running the cellular metabolism to these tissue sites. So, and at the tissue sites where you have more of carbon dioxide, because cells are respirating themselves, so they have more of carbon dioxide. At the tissue site, they will bind to carbon dioxide and come back to lungs and release the carbon dioxide. So, are interchangeably binding to carbon dioxide and oxygen depending on the concentration at the site. Now, if you look at this graph here, this is a very important graph. First, let's just look at this line and this line, or the first and the second. The first line is for myoglobin and the second line is for hemoglobin. What it says is at what oxygen partial pressure, how much it is getting saturated with the oxygen. So what it means is, so you can see it's in a sigmoidal nature. It means how much oxygen it is binding and how much it can hold. So up till 20 partial pressure, you can see that it can hold up to zero point 0.8. It still has zero point 0.8 saturation. It can still go further and bind, but only up to a certain degree. So what it means to be sigmoidal is, you can, let's say it's just in terms of binding, so you only bind to a certain extent, and beyond that you do not bind. So you have a line going up, but after a certain point of time you reach a saturation, and this kind of curve is called uh, sigmoidal curve. So here, the point to refer is, if you have a 20 mmHg pressure, at that point you have myoglobin binding to the oxygen, and the hemoglobin also binding to the carbon dioxide. But this reaches saturation faster than the hemoglobin. And that's why hemoglobin, because hemoglobin has four sides to bind to oxygen, it reaches saturation little later than the myoglobin. Now, you go to third. Third is where you have hemoglobin, but you have something called BPG, bisphosphoglycerate. Bisphosphoglycerate will tell the hemoglobin to reduce its binding to oxygen. And as a result, the saturation will go down, the binding will decrease. And if there is carbon dioxide, which is what happens in the tissue level, so at the tissue site where carbon dioxide is more, its saturation level goes very down with carbon oxygen. It releases the oxygen here and then binds to carbon dioxide here, because the carbon dioxide pressure is much higher than the oxygen pressure. So it releases the oxygen and binds to the carbon dioxide. So if you have BPG or CO2, they reduce the binding of hemoglobin to oxygen. And this is how the oxygen is transported in the body. Okay, the next kind of interaction, or the next function, is cell surface receptor. This we had discussed in the previous sessions. So these are proteins again present on the cell surface in the form of receptors, and they will interact with molecules present in the extracellular matrix. We have told this not only on the extracellular surface, but they can also interact with the proteins which are present on the intracellular membrane or intracellular location. And the cell surface themselves can be taken into the cell and recycled back and delivered to this original position by vesicular trafficking. And we had seen this in the Golgi lecture, or in the Golgi Golgi and ribosome complex, and when we were discussing that. So just to give you a quick recap, so you have receptors here. These receptors can bind to any chemical messenger and then carry the message forward by several reactions inside the cells. Enzymes, as we have already mentioned, that they can bind and change it to some product. You have a channel, so ions will move inside the channel given upon the cue. So let's say there is just some ion that needs to be moved inside the cell. So the channel will open up and the ion will move inside. But if you have something called gated channel, that the movement or the opening of the gate will be dependent on a certain factor. Let's say it would be dependent on the binding of the ion, or on the concentration of the ion. So that will be gated. That will be like how in general case also, if you have a gate, you decide to open or close it based on some cue. Here also, this receptor or this protein here will open only if it receives a cue that it has to open to let the ions inside the cell. And then you have cell identity markers. So you have these kind of proteins here, which will have these kind of some attached biomolecule with which you can identify, okay, this is this kind of cell because this has this kind of protein marker. And then you have cell adhesion molecules. So you need the cells to be attached to one another. So these kind of molecules will attach one cell to another. So these are the different kind of proteins on the cell surface that will enable the several functionalities of the cells. The next is protein function is phagocytosis, which means that when you have an external particle or anything that you need to entrap and digest, then your proteins play an important role there as well. So, processes by which pathogens or unwanted tissue debris are scavenged from the host body is called phagocytosis. And generally, macrophage cells take part in this, where they extend their pseudopodia around the foreign particle and entrap them into a phagosome and take it up into the body. And then they fuse it with the lysosome. Lysosome has enzymes which will then digest this material. And there are several proteins here which will form this vesicle. There are proteins in the lysosome which will lead to the digestion of the material. The next in line is inflammation. Inflammation is again a response to any kind of invasions or infections. So that also macrophages take part. Macrophages, for example, if they're exposed to bacterial components or interferons, activate to produce wide array of inflammatory molecules to again inhibit or kill the pathogen. And they produce ROS or the reactive oxygen species, which neutralize the or oxidize the pathogenic organism. Basically, all these are just to evade the or to let the bacterium get trapped and not evade the immune system. So they reduce the microbial burden in the host, but excess production of this. So remember, like you mentioned here, that they have produced this ROS. ROS are oxidative agents. So if you have too much of ROS in the body, that could also be an issue to the cells, because the cells, if they are too much of number, they will start attacking the host cells itself, and that would be a problem for the host cell. But in normal scenario, if everything is okay, the RO is a mode of destructing the bacterial cells that enter the cells, host cells. So if you see, for example, macrophages or monocytes or Kupffer cells, they are the immune response cells. They will have these different kind of receptors on their membrane that will bind to the bacterial antigens. And then they will have these different these different proteins levels. Either they go up or go down, and because of which these cells will come, the bacterial cells will be either killed off or ingested and then killed off. All these are different reactions or different chain of reactions that will happen once the receptor on the membrane of these cells bind to any foreign invaded particle. The other function of protein is to tissue repair or remodeling. This is especially it's important in the case of, for example, you have an injury and the skin needs to be regrafted, or the skin has to heal itself. At that point also, you have proteins which are very important. For example, uh, so what happens generally is, if you have an injury, you have accumulation of these dead and apoptotic cells and cellular debris, and these are phagocytized by macrophages. Now, at the wound bed means at the base of the wound, you have macrophages. They will, because they are already secreting factors which are responsible for destructing the invasion or destructing the causal organisms. They will also secrete something called transforming growth factor or platelet derived growth factor or fibroblast growth factor. Now, name suggests that they are related to collagens and fibroblast cells. So collagen is what makes up your extracellular matrix. So extracellular matrix gives a boundary for the cell to regenerate. And you have fibroblast cells which are attaching to each other and growing in each other. So your that wound site is getting healed by all these new cells which are coming up and forming layers. And then they also secrete MMPs to degrade the collagen, which has fibroblasts and endothelium. So first they will give the structural layer, then they will generate something called matrix metalloproteinases, kill the matrix, break off the matrix, allow the fibroblasts to move further and create new blast. So there will be angiogenesis, and you have this new skin coming up. So all these are very natural processes. You can see it in any of your injury site that you will have a first you have a clot, then you will slowly you will get your skin back there. All these are important because all these are happening because of these important proteins in your cells. So just to give an overview here, if you have, let's say, acute damage, you have these monocytes and CCR2 plus, they will be giving. So this is the inflammatory stage where they will start first try to stop the invasion, and then they will start secreting these IGF, TGF, IL that will start in wound healing. And this will also be the case if you have a chronic response or chronic injury. So based on all these uh, functions, these are some questions. So the first is the pathogen cells and the unwanted cell debris are scavenged from the body through a process called what? We have just discussed it. The second is to kill the pathogen in host and the inflammatory molecules. They are activated by which of these cells? During injury or wound, platelets release a mitogen known as what? And mitogen means something that promotes mitosis, means to promote cell divisions. So basically you want more cells to come up in the site. And the last is the type of saturation curve exhibited in hemoglobin for oxygen and carbon dioxide binding. This also we have discussed. So the first was phagocytosis that we had already discussed. Second was monocytes. The other cell were Kupffer cells and the macrophages. Third was PDGFs, as we had discussed, PDGFs and transforming growth factors, and you have fibroblast growth factors, FGFs, all these. And the fourth was sigmoidal, as we had discussed about the binding of hemoglobin and myoglobin with for the carbon dioxide and oxygen. Then these are the other set of questions. So the first is a general mechanism. Is there an enzyme acts by what? So what is the way the enzyme catalyzes the reaction? Second is the factor which does not modulate the binding or oxygen to hemoglobin to produce oxyhemoglobin in lungs. Basically, which of these does not affect the binding of oxygen and hemoglobin? Third, enzyme catalyzing rearrangement of atomic grouping without altering molecular weight of or number of atoms. Basically, which of these form a just a structural rearrangement? And the last is the GT-based protein, which is involved in pinching of cladrine-coated pits during uptake of LDL cholesterol. So the first is reducing the activation energy that we have explained already. The second question, where you have pH, H, and low bisphosphoglycerate, low or high bisphosphoglycerate affects the binding. We've also seen how pH also affects the binding, but in temperature does not really affect the binding of oxygen and hemoglobin. Third is, as you remember, isomers are also something that you call just two forms, just structural rearrangement of each other. So isomerase is what the enzyme is, which does this function. And the fourth is dynamic. Now, we have discussed proteins, what is the function of protein. Now we'll discuss some assays that you can use to detect proteins. We have seen some method that detect amino acid and analyze amino acid. Now we'll see some method, or one of the method which has different forms, by which you can detect and measure these proteins. These proteins could be antibody, hormones, peptides, proteins, antigens, anything, but they are proteinaceous in nature, so you can use this technique, which is called ELISA, or enzyme linked immunosorbent assay, to detect their concentration. And how does it work? What is the principle of it working is? So it will yield quantitative results. So the idea is to quantitate, so this will be a quantitative result here, uh, which is based on the separation of non-specific and specific interactions that take place, and you do it via serial binding to solid surface. What you mean by that is that you will have a surface where you will have something attached, either a antibody or an antigen, and then you have your desired compound, and then you use some additional detecting compound that first detects your antigen, or first detects your desired molecule, and then gives you a quantitative visualization where you can look that how much of it has bound and how much of it is present in the sample. So I'll go over it in proper details as we go over two of these methods. But first, to understand, there are four different type of ELISAs. So the one is called direct, the second is called indirect, third is sandwich, and fourth is competitive. So we will discuss indirect and sandwich in bit more details, but I will give you an overview here along with the other two. So first is direct ELISA, where you have the, so what happens, these kind of experiments are taken or are carried in something called titer plates, micro titer plates. So you will have some plates as the base, where you will first attach your first coated layer, and then you will undergo carry the forward the several steps ahead. So whatever you seeing here as the line is basically the base of this plate. So for direct ELISA, you have your antigen bound on the plate, and you have this antibody, which is what you are using to detect. So you have taken a sample, you put it directly on the plate, it has coated whatever amount it could have coated, and then you use your antibody, because antibody binds to antigen. This antibody will be specific to the antigen. It will bind to the antigen, and this antibody is also like, it has a chromogenic substrate with it. So if you see the antibody here, it will have this thing here, uh, or enzyme to it, connected to it. So basically, you will have an enzyme connected to it, which will and give you a colored substrate, so colored product. So what you have, let's say it is a horseradish peroxidase. So you will give it a substrate on which horseradish peroxidase can act, and that will give rise to a product that will give you a color, and that color will tell you that the reaction has taken place or not. And you can see that color has happened, so you know the reaction has gone here. But a direct ELISA means that you use only the antibody which is directly used against the antigen here. Now you have an indirect. So in indirect, the first step remains the same, that you have an antigen which is bound to the antibody, but the antibody which is actually used for detection is not the antibody that you have generated against, specifically against the antigen. This is something called a secondary antibody, which is what is conjugated with an enzyme, which will be giving us a chromogenic substrate to a chromogenic product. So the indirect means that you are using a second antibody here. Now sandwich, the name only gives you an idea that it is a second sandwich. There is something in between two layers. So the, because we are trying to detect the antigen here, you have first something called capture antibody. So instead of putting it on the plate directly, you have first put the antibody, which will be the capture antibody, because that will bind to the antigen here. And then you have the specific antibody, and then you have this detector antibody here, which will be the one that will give you the color. And then you have competitive ELISA, where you use inhibitor antigen. So you try to see how much antigen is binding by using inhibitor or competitive binding, how much of these get displaced by this competitor, and then you see how many free antigen are there, and you try to understand what could have been the level of original amount of antigen in the sample. So this is how you carried out ELISAs. So we'll discuss indirect ELISAs in detail. So as we have mentioned, indirect ELISAs means you are detecting the antigen in your sample, or the concentration of protein in your sample, by using a secondary antibody conjugated with an enzyme. So as I have mentioned, the antigen is attached on the wells of the microtiter plate, and the sample containing the antibody is added to the antigen coating well, and the primary. So, yeah, so after every round here, you will see that we will wash away, so that the ones which are not bound, let's say, they are removed. This is to remove noise. So let's say you have, just arbitrarily, just imagine that you have 10 antigens on the plate, and your sample has some 20 antibodies. Now your 20 antibodies will still bind to the 10 antigens that are there. Now there are 20 antibodies already in the plate, and you now put the secondary antibody. Secondary antibody's role is to bind to the antibody. You have given 20 antibody, no matter or irrespective of the fact that the 20 matter, irrespective of the fact that your, so I'll just go over this once again just to explain it. So you have, why do you need to wash during steps of ELISA? So you have antigens, let's say very arbitrarily, you have 10 antigens on a plate, and you, that means you have 10 binding sites for that antibody on your plate, and then you have added your samples, and your samples happen to have 20 of antibodies. Now you put these antibodies from your samples into the titer plate, and because there are 10 binding sites, only 10 of these antibodies will bind, but you have 10 extra antibodies which are not bound to any antigen, but they are still there in the microtiter plate. Now you use the secondary antibody, which is what you will use to see if you have any reaction in the plate or not. The secondary antibody will recognize the antibodies in your plate, no matter if it is bound to an antigen or not. So it will bound to all the 20 antibodies in the plate, and then it will give you an yield or a signal where you have given the substrate and you will get the chromogenic product. So when you get the result, you will, it seems to you that there are 20 antibodies which are bound, but the reality was that there were only 10 antibodies which were bound to the antigen. So there were 10, the original result that you should have got was 10 and not 20. So that's why you have a washing step after every step in ELISA, so that you remove these unwanted or unbounded biomolecules. So and so how do you calculate the final absorbance, or how do you calculate the yield? Is when you have this signal as the colored product of chromogenic product, you use it by you measure the absorbance value by spectrometry. So that was the brief. So let's go over steps one by one and try to see how does actually take place. So the first thing is coating. Coating is where you add the first layer. So you have these antigen solutions, and you put it overnight on the microtiter plate, so that it goes and binds to the what do you call, how much ever antigen was there, it goes and binds to the plate. Now you have something called blocking. So you block with 1% BSA. And what do you block? What is the blocking thing? The blocking thing is, on the microtiter plate, whatever site has been left unbound by the antigen is bound by the this BSA here. And why is that? Because you don't want anything else to go and bind there, and you block these regions so that there is no noise from these regions. So you block these unbounded sites. Now you then prepare antibody dilutions, and you then add these dilutions onto the main plate. Now what is antibody dilution? Antibody dilutions means you are serially reducing the concentration to see the change in the binding. And why do you need to do that? You need to find the antigen and antibody complex at the proper concentration. So you have more of antigen, less of antibody, it will not bind properly. You have more of antibody and antigen, it will, there can be a possibility of much more binding than it should actually be. So let's say, just try to imagine there is an antibody and there's an antigen. Now, this inherently has a very poor binding. This antigen cannot bind, antibody cannot bind to the antigen. But if you give too much of antibodies to this antigen, all will go and bind, and it will seem to you that there is much more binding happening between the antibody and the antigen. But in reality, it was very less because of the fact that you had given much more concentration. You see a higher binding. So to avoid this kind of confusion, you do a serial increase or decrease in the concentration, and you check if the absorbance values is changing accordingly. And how do you do a serial dilution? So there are two tables here, and there is this is how a microtiter plate looks. So this is how the color gradation will look. So we'll go over this in a bit. And one of the most important things to do or to know here is whatever test you do on ELISA plates, you always do it in triplicates, because if you can get one result as erroneously, if you do it just one time, you can't be sure of that result. But if you get the same kind of result, even if it deviates from what you would expect, but you do it three times and you get three times the same result, the chances of it being due to an error or due to it being the case of just a chance discovery becomes very less. So that's why you do it uh, at least triplicate. And statistic, so what do you call this in science, is you call them to be statistically significant results. Whatever you see, you are quite confident of reporting that result. Okay, so let's try to understand serial dilutions. So let's say you have this as your antibody, and when it came to you, it came with some 1 microgram per ml. So you have very high concentration. If you take 1 ml of this vial or 1 ml of this solution, you will have 1 microgram of the antibody, for example, in this situation. Now you take 10 of this, 10 microliter, and you dissolve it in 990 microliter buffer. So what is the total volume? The total volume is 1000. So let's do this calculation inside so that you get understand it. So the total volume is 1000. So you have 1000 as the total volume. In this 1000, you have 10 parts of this antibody, because you just took 10 microliters. So in 1000 microliter, you have 10 microliters. So if you just put 10 here, so how much is your actual concentration of the antibody? It is 0.01. 0.01. So here the concentration has now become 0.01 of what you originally started with. And what did I start with? I started with 1 microgram per ml. So now it had become 10,000 picogram per ml. The next step is, so how do you serially dilute it? You always keep your total concentration or total volume constant. So if it is 1000, you keep it 1000 all across and keep changing your antibody or your whatever molecule you're looking at. That concentration keeps changing, but the total volume remains same. So accordingly, you will change the buffer as well. So now you had this 1000 ml here with 10,000 picogram per ml. Now you take 200 from this, and you have to put it in this third vial. Third vial should also add, or second vial should also add up to 1000. So if you're taking 200 of the first of the antibody from the first tube, then you have to add 800 here, so that 200 plus 800 makes 1000. So now in this tube you have 1000 again, but this time you have 200 microliter. So you have 1 by 5th of this concentration. So whatever concentration was here, it becomes 1 by 5th. Not what was started with, but what was in the previous tube. In the previous tube, it was 10,000 picogram per microliter. You have now reduced it 5 times. So it becomes 2,000 picogram per microliter. And now you so it became reduced 5 times. Now you keep reducing it half times every time. How do you do that? You take equal part buffer and equal part of your solution. So now it starts reducing by half each time. So You take 500 from here and you take 500 from here, it becomes 500 in total 1000. That means it becomes half. So half of this concentration is 1000. And serially now half, it keeps reducing half. So it becomes from 1000 to 500 to 250 to 125. Half of that is 6 to 62.5, 31.25, and respectively keeps decreasing to 56, 15.6, 7.8, 3.8, and lastly it reduces to even further. But you don't need very less, but you need up to this much part. And here it is important to do this because that will give you a standard curve of what should be the ideal absorbance that you should observe for this sample, given that you reduce it at a certain concentration. And then you use your compound and try to see what you get. We'll come to that. And like I said, this is how you, so see, as and when your product is getting, or your antibody is reducing, for example here, the binding also is getting reduced. So the intensity of the color is getting reduced, and you can see how the color gets differentiated in each of the triplicates here. Let's consider these to be triplicates, or at least double. So you see the colors are reducing in doubles here. And so this is one, this is the other. So 1 is to 100 means what is the level of dilution. So this could be either by concentration or by the level of dilution you're using. So let's say in the first well, you use 100, 1 is to 100. 1 is to 100 means one part in the total 100. So 2 by 200, because you are adding 198 buffer, so the total volume of the tube is 200. In that, 2 microliter is antibody. So 2 by 200 is 1 by 100. 1 by 100 is 1 is to 100 dilution. Now what happens is you take 20 from this and you respectively reduce the buffer and keeping 200 constant volume. So now it becomes 20 by 10, 200. So 1 by 10. So whatever was the dilution there now reduces further 10 times or increases further 10 times. So the dilution now becomes 1 is to 10,000. And you keep continuing this for the third one also. Now it again becomes 10 times more diluted. So it becomes 1 is to 10,000. Now in the fourth one, you take 100 of this and 100 of this. So now you actually double it. So whatever your dilution factor was now goes double, because you have taken equal part of buffer and equal part of your antibody. So now the dilution increases to 1 is to 20,000. And then you continue this for the remaining vials. So you keep increasing them. So 1 is to 20,000 dilution, 1 is to 40,000, 1 is to 80, 1 lakh 60, and 1 lakh 30. Now what do you want to understand from is, so as and when you are diluting, you're reducing the number of antibody, and you are still checking if at reduced volume also, or if at reduced concentration also your antibody binds very high, you still get good intensity of color. It means the antibody really binds well to the antigen. Here it was bound to bind well to the antigen. But what if you find good binding here as well? And it is also very obvious that if you reduce it to a very low number, it will not bind. But that is your checkpoint, that you have very high intensity here and very low intensity here. And this is where you try to see how good it is binding to the antigen. So once you are done with this, so let's go back to what we had, where we had stopped. If I just go back 2 slides, we had prepared the antibody dilution and we had added it to the tubes, or to the plates. Now we will wash the plate, and I had mentioned the importance of washing, because just now I said that antibodies will be added to the plate so that they can bind to antigen. I will now wash it so that the antibodies which did not bind to the antigens are washed off. And then I will add the secondary antibody which will have the enzyme connected to it or enzyme bound to it. And again I will wash that off so that that does not give me a wrong signal. And once that is done, then I will give it substrate so that the enzyme can change the substrate to a chromogenic product, and I can get the color as I just showed you in the previous slide, or something like this. And then I can take this plate into a spectrophotometer, take its reading, get the absorbance value. Now once I have the absorbance value, I will have something like this. Now you can either have this curve in an increasing manner or a decreasing manner. Here it will be in a decreasing manner because I kept reducing the concentration of antibody as I went ahead in the plates. So what do you do once you have the absorbance values? So you take the mean absorbance for each set of sample, Subtract the background. The background will be the one in which you haven't added anything. It's just the uh, buffers which do not have a colored which are where the chances of getting colored products is zero there. So you have the background absorbance from each mean. You draw a calibration by plotting the standard protein concentration against the absorbance that you have found to produce a typical standard protein curve. Then you use an regression analysis, and you get that equation. And in that equation, you put your absorbance and you get the concentration of your unknown sample. Let's try to understand it. Now, let's say this was your absorbance values and this was your concentration. And how do you see it? So you have an absorbance here, you know the concentration, absorbance here, you know the concentration. So you take the concentration on the x-axis and you take the absorbance on the y-axis. And of course, as your chromogenic substrate intensity goes down, your absorbance will also go down. So your absorbance keeps going down. At the end of the thing, and I've done your experiment, you have all these results compiled like this. Now you want to see what kind of equation explains is the best. So you will try to fit an equation which gives you the fit of this line. Something like, let's say you have a straight line, and you must have read that a straight line is given by the equation y is equal to mx plus c. I'll write it for you here so that it's easy for you to understand. So you have y is equal to mx plus c. So if you have a straight line, something like this, so for that you have y. So for that means that each y value here will be determined by the slope of this line at the x value and the c, the intercept here. So let's say you want to find a y value given an x value. You know this value, what is the slope of that line and what is the intercept of the line. You can find the value of the y. So this is some kind of equation. This is called linear equation, and that's why we are doing linear regression here. If we are using linear equations, and we're trying to find that what kind of equation possibly can come up with this equation. So what do? You take 2 points here and you try to find the difference between the x and the y's, try to get the slope, try to get the intercept, and try to come up with the equation that can explain it the best. And you check that how well that equation can define the values that you are getting. So this is what you call linear regression. You fit a line here. So how will you read this? By any y value given an x value will have a slope of minus 3.4. So the reduction is the slope, or the downward slope is minus 3.34, and the intercept is 1.3. So if you extend it even further, you get an intercept. The line will cross, or it will intersect the y-axis at 1.322. And r square is how fit this line is. So the fit is around 98.984, which in percentage is 98.4, which means it is a quite high, well fitted line. So now you have these values, now you have this equation. Now this was for a standard, now this was for a known protein concentration. You knew that this much you have reduced, and that's why you have this much of concentration differences. But you then have given everything same except for the concentration of your sample, so a certain absorbance. So you have your y value and you have everything available except for the x value, which is the concentration of your sample. So you just take your absorbance, take all these values, put your absorbance value here, and see what was the concentration of your sample. And that's how you try to get to the answer of your sample concentration. So this was about indirect ELISA. So ELISAs work on the same principle. Even in sandwich ELISA, at the end you will be doing the same thing, but the change is in the process of getting to the how do you capture the sample. So for example, sandwich ELISA, it will be it will be generally used to detect the presence of antigen. So you start with the coating of an antibody, and then the sample will be added here. and sample will be the one which will contain the antigen. So there we will detecting antigen, here we will here we are detecting antigen, there will be we will be detecting antibodies. And then again the same thing. So you have your antibodies which will bind to the antigen and an antibody which will bind to the antibody. So if we go step by step, the first step is coating. Again, the first step in sandwich ELISA is coating of the capture antibody. So you have the capture antibody on the wells. Then you block these remaining sites which have not bound to any of these sites. Then you aspirate the sample. Basically, you then add the antigen. You first add the antigen, keep it for some time, let it bind, and then you remove the antigens which are unbound by washing them. And then you use the detectors. So that is called detection. This is or something you call working detector. So you have your detection antibodies, you incubate them and then you remove them, and then you add your substrate, and then you check it at your absorb or the wavelength that you should check for. And also you, if you have to reduce the background absorbance, you do that as well, and then you finally come to some graph like this. Now this the previous one was downward slope. This is in the increasing order because we have plotted in the increasing order of the concentration. So that's why this curve is looking this side, but the idea still remains the same, that you have a standard curve here. You get your equation from this, and once you get your equation from this, then you put your absorbance for your sample, and from that you calculate the concentration of your sample. So based on these that we have just done, we these are the questions again from previous year's assignment, so we'll try to solve them. Okay, the first is the molecular technique used to identify an antigen in the bioecological fluid. Second is the purpose of blocking solution in ELISA is which of these. Third is what is the principle, working principle of ELISA. And the fourth is what is a primary antibody. So yeah, the first one was very easy, but just to give an idea, ELISA is the technique that you use to identify antigen. Chromatography also we have discussed in this session, where we have seen that it is used for separating different components of a sample. Southern and northern blotting are related to DNA and RNA blots, or how you transfer DNAs, or how do you detect DNA and RNA using a membrane. So that was why I gave this option here. The second was, why do you use blocking solution? So the answer is to block the remaining protein binding sites. For stopping enzyme reaction, what you do is you use a substrate. You give substrate, and you after a certain point of time you take the absorbance. To block all the binding sites, you don't want that to be happening, because first you want the binding sites to remain open so that it binds to its respective antigen or antibody. And to prevent the specific binding, that just outrules the entire principle of ELISA, which works on the component of specific binding. So that was the third question. What is the basics of ELISA? Is it antigen antibody neutralization? No, because we are just trying to find if it is binding, first if it is binding, and second how much it is binding. Nothing to do with the neutralization. And fourth was, what is the primary antibody? So any time you call primary antibody, it is the first antibody that you use to detect, either in the capture antibody or in the indirect ELISA or the direct ELISA or wherever you're using it. So with that, this session is about to end, and we don't have any other questions to discuss.